it's been a pleasure and an honor to be here today. I know it's the end of the day, so I'm going to try to not take too much time, and you can actually stop me. But I thought maybe I'll take a slightly different perspective from the morning keynote, although it gets down to the same issues, to think through what the relevant categories may be and what we need to understand before we get to treatment, especially when we talk about biological treatments. So I'm going to put that up for a second just to have done my disclosures. So I'm going to take a step back and say that we're talking about autism here, but autism is not the only neurodevelopmental condition kids get. And in fact, there are conditions that have similar prevalence or somewhat higher prevalence that have overlapping symptomatology and service needs. They tend to all be on the expensive side in terms of lifetime cost. And the truth is we have not changed the trajectories of these disorders, even when we have successful treatments. So the adult outcomes of kids who have neurodevelopmental conditions have relatively stayed stable for some time. So, and the other thing that we are struggling with a little bit in this space is that the emergence of understandings in biological treatments, in underlying biologies, have not yet translated to successful treatment. So most of the medications and technologies we use have not come through the translational route. So it's not surprising we haven't actually changed trajectories if we're not treating the underlying pathophysiology. And there are all kinds of reasons why this has been delayed, and Joe mentioned some, and there is quite a few more, but the three I'm going to focus a bit more is, well, the limited number of targets that we have confidence in, the limited number of targets that are actually druggable or maybe good intervention targets, and then a whole group of challenges that have to do with precision medicine. So methodological challenges around how you match the right intervention for the right kid at the right time, because these are developmental conditions. And the lesion, if you like, I'm a neurologist, and I think in terms of lesions, is not stable across the lifespan, changes, right? So not only you need a targeted treatment to the kid's original biology, but you need to have some ability to adjust it to the compensation that happens sometimes strategically by the brain and sometimes because of stochastic events, right? So you need to be able to be targeting the lesion the child has now, not the lesion that originally produced the developmental difference. So a bit complicated. So I usually put a slide like that up to remind us of these two phenomena. One is the heterogeneity problem, and the other one is the pleiotropy problem. So this is a family that I see, and they have given me permission to use their picture. And I usually use it to highlight a couple of problems. So these are three SIBs. They share some genetic variants, and they also have some de novos on top of their inherited variant. The first one is a boy with borderline intellectual disability. He's not doing too badly, but he needs educational supports. Has severe anxiety and internalizing symptoms. The second one is a girl who is in the gifted range in terms of her academic abilities and her cognitive abilities. She is experiencing right now gender dysphoria. She has a diagnosis of ADHD, but not a diagnosis of ASD. And then the little one is the little guy who has autism, classical autism, has a relatively high split in his IQ with very high verbal skills, but some difficulties in his kind of non-verbal abilities. He's the kind of kid who learned how to speak Russian at the age of 3. There is nobody Russian in the family. He just liked how it sounded on YouTube. And I am told by Russian speakers in the group that he's close to fluent. On the other hand, he has severe externalizing behaviors. His mom is black and blue. So you can see this is a family with shared genetic background and quite a bit of a shared variant load in terms of the things that we think contribute to the neurodevelopmental disorders, but very, very different presentations. So if you look at my bubble graph, and we all have a version of this bubble graph on the other side, I don't know if you can see perfect. I'm a child neurologist, and I don't only see 2-year-olds. In fact, I almost never see 2-year-olds, because the majority of the two-year-olds get diagnosed in practice developmental pediatrics and psychology practices where I am. And so if you only see 2-year-olds, you see lots of kids who just have autism. But I see kids that are 15, 16, 17, and they almost never only have autism, right? So it's not just that there are co-occurring conditions, it's that autism almost never exists in isolation the older you actually sample these kids. So what does that mean? So what does it mean for our diagnostic constructs if we almost never see them in isolation, right? If they always coexist with other neurodevelopmental conditions. So I'm not going to go through you guys are experts in a lot of these co-occurring conditions, but the idea is that if this diagnosis coexist in the majority of the kids, is it possible that the diagnostic constructs as we articulated them are not actually mapping into distinct biological categories? So what is the validation for autism, right? So there were field studies, people observed a whole bunch of kids who had social deficits and repetitive behaviors. They have certain flavors, although those of us who see autism, there are many different flavors, right, that we recognize as autism. But when did we go through the exercise to say this is a valid construct? It has distinct biology, it has shared biology and distinct from other conditions. So if you allow me, for the purposes of this discussion, and I'm not taking autism away, I promise, I'm gonna start from the position that in fact there is no such thing. So that the conditions as we describe them and as we develop the criteria for don't actually exist as distinct biological entities, and therefore our ability to translate our understanding of these conditions into effective treatments is very problematic, because not only we see many different conditions under autism, let's say, like Joe mentioned in the morning, but they're actually overlapping with other conditions. So there is no specificity and no sensitivity to the construct we developed, right? Now, I know it's an extreme statement, right? So you can take a few steps back from it, but challenge yourself to think that this may be potentially true, right? So if that is true, then what is it that we're doing translational work in, right? So Joe talked quite a bit about genetics. I'm not going to talk about genetics. The only thing I'm going to talk about here is not only that a lot of these genes are found across multiple developmental conditions. And when I think about neurodevelopmental disorders, I don't just think about the differential of autism versus DD. I think about the ADHD kid who has social deficits. I think about the OCD child that has social deficits, and so on, right? So these genetic variants, we pick them up in various conditions. And so we have the large genomic heterogeneity, we have the large phenotypic heterogeneity, and we don't have one-to-one mapping anywhere, right? And I think that's where we are. So we can articulate our distress about this in various ways, but the truth is, this is kind of where we are, with few exceptions. We have certain rare spaces where we have some clarity, but for the majority of the kids, we don't have clarity on how we map biology to behavior within autism and across neurodevelopmental conditions. So how we're thinking a bit differently, what I'm going to show you now, is that we're taking a step back and saying, let's pretend there is no validity to our diagnostic constructs. We all understand neurodevelopmental difference, so we accept that. But where exactly we put the boundaries, let's take it back and say we may have been wrong. And if that's the case, then there must be another way to classify the neurodevelopmental spectrum in a way that produces more biologically homogeneous conditions. So that's the premise, right? So I'm going to use this premise to think about the lumping versus the splitting. Of course, there's no straightforward answer. You lump and you split, right? But the question is, you split around which boundaries and you lump across what conditions? Okay, so I'm going to use a single network to make the case for some of these things. It's not the only thing we do, but I think it makes a decent case for the kind of questions we're asking. So this is the Province of Ontario Neurodevelopmental Disorders Network that includes a clinical trials network that's embedded in a large biomarker core across neurodevelopmental conditions, so that we can start understanding what's shared and what's unique across these conditions, or have the chance to start from scratch if we needed to do so. So you'll see the institutions that are linked in Canada across this, but the idea is we recruit kids with autism and ADHD and OCD and intellectual disability and any genetic syndrome that has a developmental phenotype, whether we can label it or not, right? And so kids come in and they contribute sample for genomics, and they get behavior characterized across common dimensions, but also experimental paradigms around cognition. We scan as many of these kids as we can, so we have both structural and functional neuroimaging in a large number of those kids, and that's the kind of data I'm going to show you in a sec. And in the last couple of years, we also added immune and endocrine platforms to look at features that may help with classification across the neurodevelopmental spectrum that may not quite fit across these diagnostic lines. And at the same time, we have a large IPSL core, that I'm not going to talk about today. And we have a large mouse model core that I'm going to talk a little bit about today, just to show you how we use mice in this exercise. And of course, at the end of the day, you can imagine this becomes a computational exercise. And there's lots of you here with computational expertise. So there is a bit of a computational core. So we are up to 3,000 kids enrolled right now. It sounds like a good number. It's not a good enough number. But I'm going to start showing you a little bit of data from this cohort to give you an idea of where we stand. So first of all, genomics I'm not going to speak about, except to say that you heard about some initiatives this morning and right now. Another one to think about is the Missing Initiative, which is a collaboration between Canadian networks and Stitcher and Google and Verilai. Sorry. And so the only reason I'm putting it up is to remind us that none of us has enough cohorts and none of us has enough numbers. Spark probably will be the exception to the rule, but it comes at the cost of deep phenotyping. So the idea of combining or putting our data sets in the public domain for people from anywhere in the world to be able to interrogate is critical. And so all of our genomics data from POND goes into Missing, and it's in the public domain as we collect it. All right, so I'm gonna just show you a couple of behavior ones. I have to admit something, and you're gonna forgive me in advance. I'm a neurologist, so my faith that behavior will work in the classification is kind of on the low side to begin with, but I'm going to show you a little bit about this. So some of the first things that we did is we tried to take domains that were supposed to be diagnostic of one condition, but not part of the criteria of another condition, and see if in fact they separate the diagnostic categories. So very, very simple. You take a task like this, so the reading the mind in the eyes task. Some of you talked quite a bit today and during the posters about empathic accuracy. And you just evaluate how kids do across the different conditions. And you'll see that autism obviously did the worst, but ADHD did pretty badly also. And then people were like, well, they're doing badly, but they're doing badly for the wrong reason. So kids with autism have a social deficit, so they're finding hard items. So in this particular measure, you can separate hard from easy items. And so the prediction was that kids with autism will fail the hard items but get the easy items, where kids with ADHD will have random response rate because they're not paying attention, and that's why they're scoring poorly. And it's not the case. So if you look at easy versus hard items, everybody's doing poorly in this task in the hard items, but they're not separable. And then if you look at easy, you get the same pattern that we got before. And then the psychologists said, this you're too crude about this. Valence matters. You need to look at valence. So separate positive out of negative emotions and neutral phases. The psychologist in the room knows that there's no such thing as a neutral phase. when we are completely flat, the interpretation that people take of a flat face depends on a whole variety of things, but it's almost never interpreted as neutral. We usually have an expression. And so depending on the condition, some people will, or the style, the cognitive style, some people will always interpret a neutral face as a negative face, as an ambivalent face, while others not. But the point is the pattern was the same for neutral. But what you'll see is that for negative faces, when you have a lot of attention, very salient, there was absolutely no difference between, in fact, all the conditions that we looked at. And for happy faces, for positive emotions, autism and ADHD were not discriminated at all. So there's some early data to suggest that maybe some of these domains do not separate the diagnostic categories, making a little bit of an argument for lumping there. Now, we just by starting at the same place does not mean you're going to end up in the same place, because remember, you have all these co-occurring conditions and various biologies that may predict different trajectories, right? And so we have started looking at where the kids end up in terms of adaptive function. So lots of you mentioned the ABAS today. We use the ABAS also in the consortium. And so the only thing I put this there for is to show you that although they start looking pretty overlapping in terms of their social cognition when it comes to social function, in terms of leisure items or social items, everybody seems to be doing better with age. They start at different points, but they do better with age, except for the ASD girls. So the ASD girls, in our hands, start better with some skills, but they actually flat out in terms of their social adaptive function. These are norm scores, so you have to remember it's basically they're doing worse and worse relative to other typically developing girls. So you have to think about what girl phenotypes look like and why this may be harder for our autistic girls. Or caveat here, we just converted the cohort into a longitudinal cohort, so everything I'm showing you is cross-sectional, so everything goes in quotation marks when I talk about trajectories. But the one that's a bit more interesting for me for behavior before I show you imaging is when we created a factor structure of the combined autistic and ADHD symptoms across all kids who have autism and ADHD. So by that I mean we normalized the score on social repetitive inattention and hyperactivity for the total number of symptoms, the total burden of symptoms. And for each kid, and every column here is a kid, we had a social score, which is the red, a hyperactivity impulsivity score, which is the light blue, an orange score, which is restrictive behaviors, and an attention score, which is the dark blue. So the first thing I want you to see is the thickness of the band across these colors. And so you can start looking at the two blue that are the ADHD symptoms and tell me whether you think there is a lot of variability in the breadth, the width of the blue bands. So they go up and down, but in terms of the width of those bands, they look pretty much the same all across this. So in a second, I'm going to show you who has ASD and who has ADHD. And I want you again to look at this breadth of the band of the hyperactivity and attention symptoms in ASD and ADHD. They're normed, right? So this is not absolute score. This is almost like what % of your total symptomatology is accounted by symptomatology in ASD and in ADHD. And so you'll see like it almost looks like it's a default condition. So in terms of the % of their total burden, everybody has ADHD, if we believe the construct, right? If we believe the construct. And then if you look at red, which is the social, clearly the kids who are autistic have overall more social symptomatology. But I want you to look at these kids here and these kids here, and I can tell you that they have the SEQ, these are SEQ scores, are higher than the mean of the ASD group. And we went back and had the expert clinicians use their PICs and the KSADs and their ADOSs and tell us whether these kids are autistic, and they claim they're not, right? But their continuous measures not quite continuous measure for the SEQ, would suggest otherwise. They would suggest that they have significant autistic-like social impairment. Now, for those of you who like to look at girls and boys, and following the Simon's announcement about the sex differences RFA, you can see that there are some subtle differences in boys and girls, with boys with ADHD having the majority of the social burden which is kind of interesting, but for what's worth. So all of this to say we're not getting very nice separation, even when we just looked at our diagnostic symptoms across these conditions, even when we have these expert groups. These are not community diagnoses. These are expert diagnoses with all your standard instruments and all the ridiculous stuff we do because we believe in these constructs and we spend all these hours trying to get the exactly correct diagnosis, right? Yes?
The measures are parent-reported?
So these ones are all continuous, yes. So they are the SWAN and the SEQ.
So they're both parent-reported? So
yes.
Could that be also related to the fact that it's the same reporter?
It's the same what?
The same person reporting on both measures.
But these are different kids.
No, but for each child, you've got different instruments.
For the same, yes, yes. All right, so this is behavior, but I'm a bit more interested in what happens in the intermediate phenotypes. So we heard about genetics in the morning. That's for me, this is your blueprint, like it's your etiology, it's your original hit. But then there is some common effect that has happened after multiple steps of whatever the black box is, whether they're other genetic finding, other genomic variation in your genomic soup, or whether they're environmental stuff, or stochastic events, because we forget about those, that actually put you on a trajectory, and your brain should capture a little bit the trajectory that you've been on. So I'm going to show you a little bit on structural and a bit on functional across these disorders, and then we can think where we are sitting on this splitting versus lumping. All right, so this is the first one we put out, and this was just a straightforward DTI study looking at structural connectivity across these conditions. You'll basically see that the ASD and ADHD groups, this is the only area that we found that was different across the groups, and it's the posterior splenium. It's a big bundle, it makes sense. It's not a rocket science of anything, but you'll see that the neurodevelopmental groups look much more similar than the controls. And then if we looked at this and we're like, okay, fine, but it should correlate with their core symptom domains. So it's a big bundle, connectivity bundle, but it should correlate to something that gave them the diagnosis. So we looked at whether this relates to social symptoms or repetitive behaviors or inattention or hyperactivity, and it does not relate to any of those things. The only thing it relates to is your total adaptive score on your favorite ABAS. So basically, the brain seems to understand if you're doing well or not well, but it doesn't read your DSM or your ICD-10 or whatever you're using, right? So it does know, it does register whether somebody's doing well or not, but it doesn't seem to be mapping down to those things that we think are important necessarily. Now, if you do, you don't have to actually be able to decipher this, but basically these are two-way comparisons, so ASD versus ADHD, ASD versus OCD, OCD versus ADHD, and so on. So the one column that's not there is ASD versus ADHD, because in fact we could not find differences between ASD and ADHD in this particular cohort up to this point. So one way to interpret this is that these neurodevelopmental conditions are their one thing, and you should lump them. The other way to interpret this, though, in the original slide I showed you on this paper, is look at my error bar. So by no means this is one condition, right? My error bars are enormous. It's just that using the diagnostic label does not help me clarify the heterogeneity. Does that make sense? So far, for this paper. So I don't get many differences across groups, but clearly I have a lot of variability. So the suspicion was there that there are biologically homogeneous groups. We just could not capture them by using the diagnostic classification. And we have done this multiple times. I'm not going to show you now. So this is a paper where we looked at structural cortical thickness, and we had a very, very similar result. And then we were like, okay, maybe structural doesn't capture it. It's too crude. Let's look at functional. So this is resting state connectivity. And we use graph theory. For the sake of time, I'm not going to go through methods, but I'm very happy to answer questions about methods after. But we use graph theory to create properties of networks for the resting state network. And all I want you to see here, I know it's a busy slide, you get combined NDD group is blue and red ESTD. So first of all, I want you to notice that we don't have significant differences, but although we have trends that people have reported before. In our cohort, it didn't reach statistical difference, even the subcortical hyperconnectivity that's well replicated now. But then I want you to look at the pattern from ASD to ADHD to OCD and tell me whether you think the pattern looks different. So I can tell you statistically they're not different. But for the sake of not having enough samples, even to explore potential patterns that are not that we're not statistically powered to detect, we can find major differences across these networks. The only thing we find consistently that makes sense to the psychiatrist in the room and the psychologist is that OCD spares a little bit of posterior brain. It's mostly frontostriatal, but when you look frontostriatal across, you get it in all conditions. So then we're starting to get a little bit smarter and say, okay, we're not finding main differences, but my God, look at my error bars. Huge error bars. What does it mean to have to compare these means, right? When your error bars are that large, can we start clustering this data to figure out whether there are biologically homogeneous groups? And that was the first attempt, and it's pretty basic. It's K-means clustering, but basically two cluster solution worked for this one best. What's important is not how many clusters. What's important, because we only did cluster resting state. Obviously, resting state is not a diagnostic feature, so it's not enough on its own to tell you how many clusters. But what I want you to see is that there is no cluster that is diagnosis specific. So you get your ASD splitting between the groups almost evenly. One cluster is a bit more ADHD, the other cluster is a bit more OCD, and the controls are also splitting across. So when you try to cluster the biology, you do get subgroups, but you do not get them to be mapping to specific diagnostic label. So at this point, we're very basic, and we're like, okay, we need an AI group to come in here and do some sophisticated stuff, because God knows what we're looking at. And so there is now an AI core that's associated with a protocol. And this is their first paper that they put out, I think in November or December, something like that, where they took just cortical thickness and behavior on continuous domains like we did before, and clustered the data to try to see whether we can get diagnosis-specific clusters. So they got 10 clusters. It doesn't matter. Of course, it's not the right number of clusters. They only put in cortical thickness and behavior, right? But that's not the point. The point is yellow is typically developing controls, OCD is green, ADHD is light blue, and ASD is dark blue. And you'll see that you only get a subgroup of kids with autism sitting on their own as their own unique cluster. But other than that, the conditions are spreading across 10 clusters. We had the impression that this was more of a neurodevelopmental set of clusters, and this was more mixed with a lot of the controls in there. But you can see basically this is quite mixed in terms of not getting the majority of the kids fit in a diagnosis-specific cluster. You do get those last couple of ones. They're either autism or autism plus ADHD. So that's not trivial, right? Again, please be careful, because the exercise does not have all the relevant data in there to say this is the right solution, cluster solution. But so you see some kids separating out more than others, but you also see a whole lot of kids in the middle that are mixed with typically developing kids and whatever have you in their brain in terms of their structure and function. And if you look at the networks that are actually, what I didn't tell you in the resting state, clustering is that the two clusters differ in terms of social disability and attention, and always adaptive function. Adaptive function always, in our hands, seems to predict the clustering. The clustering solution always seems to end up with clusters that have differences in adaptive function, which is interesting. But in this case, when you look at brains, the kinds of networks, so the more red it is, the bigger the effect size. So the kinds of networks that you get, again, are social and attention networks, which is also interesting. All right, so this is for where we're sitting with the human data. Now, very quickly on how we use mice data. And I'm not going to show you very recent data because these guys are trying to publish the most recent paper and I don't want to mess with it. But I'll tell you what this story is about. So the story was that we're going to use mice to explore this heterogeneity, but we would start with human genetics. So these are not behavioral models of autism. There's no BTBR there. These are genetic variation that we see in kids that gets introduced into mice, and then we scan the mice because we have this ability now to scan cross species and start thinking about how one could go cross species in terms of brain structure and function. So the experiment I'm going to show you has to do with 1,500 mice and where we are now is we have scanned 1,500 mice and 60 genotypes, but I'm only going to show you the paper, the original paper from the 26 mouse models because I don't want to mess their publication. And so the idea again is that you have mice that have, and you can challenge every one of these mice, whether it's a transgenic mouse or whatever have you. There's all kinds of limitations, right? But what you get is this is a series of genetic variations that have been associated with autism and other neurodevelopmental conditions. And this is just your effect size of total brain volume from the wild type. So 0 means there's no difference in total brain volume from the wild type to the autistic mouse, please, in quotation marks. And this means that you have very small head compared to what was predicted by the wild type. And this means that you have a very big head compared to what was predicted by your wild type. So all I want you to see here is that you have some small heads and some big heads and everything in between. So if you were in your head, pretend that you calculate a mean on this and put an error bar around it, right? Think about the human data I just showed you with all the work we have done for the human data. So this tiny, tiny small effect between groups may or may not reach statistical significance with huge error bar. It is not random. If you have the genetic information, you see the structure of that error bar, right? So we don't have all the information we need for the kids, but the idea that this is just some random variation that produces this kind of large variability in the overlapping conditions is probably not true for the most part of it. It's probably predicted by the original lesion, whether this was a high penetrant genetic difference or something else that we don't know about. The other thing to notice is that I have ranked the genetic differences at the bottom the same way. The blue was total brain volume, and then the 3 orange bars are cerebellar cortex, corpus callosum, and striatum. So I want to tell you that this is not just about small brains or big brains. There is different mutations have different effects on different parts, different networks in the brain, right? So what do you do with this now, right? Because for the mouse, we have imaging, we can cluster, but I didn't even show you the clustering, but we have clustered this. Back then we had clustered this, we had come up with 3 clusters of different genomic variations that predict similar brains, and we had also clustered the brains per se, and we ended up with 3 clusters, one that was a bunch of genetic variation that mostly produced frontostriatal dysfunction, a bunch of genetic variation that mostly produced limbic and emotional dysregulation, as you would imagine, and the third one was all cerebellum, for those of you who like the cerebellum. But how do you do this in human? In this case, my first boy that I showed you there, where I have his imaging, but how do I get from there to actually understanding something about the underlying biology when I don't have all the mutations, all the genetic variation to capture? So first thing I want to say about this is we did, for the regions that are, okay, so it's mice and human, few million years of evolution in between. So again, quotation marks, but we looked at the preserved regions and tried to see whether we get any correlation between human and mouse in our cohort. So remember, we have wild types and we have autistic mutations or neurodevelopmental genomic differences in the mouse, and then we have the kids with the variety of the conditions. And there's a pretty decent correlation. And the distributions look pretty similar. So look at Newton ASD is blue and wild type is red. On the far side, you have mouse, all our mouse data, and on this side, you have all our human data, and you see the distributions are pretty similar. So we think we're capturing something that can be measured, striatum, it's all part of the brain, across species. So that's comforting. But then look at this, back to Joe's point this morning. So this is dentate gyrus. So you get mouse on top and you get human, but then we put the actual genomic differences on the y-axis, and look how much that distribution doesn't exist. So look at all the different distributions just for the dentate gyrus, depending on the genetic difference. That is the ultimate sum, right? So very similar, slight difference, small effect size, large error bar, but in fact, it is not meaningful unless you know where you're starting from. So here we're in a situation where we would be splitters, right? So I would be lumpers in terms of thinking whether my diagnostic criteria matters as much. But when it comes down to understanding the different biologies, you have to be a splitter. You can't possibly believe that that distribution is going to be helpful to you when this is what it's made of. And that's how we use the mice. So that was my parenthesis to tell you how we use the mice. And so a little bit on therapeutics. So obviously we're in a tough place for therapeutics when this is the situation. So I'm not going to talk about Joe's approach that he talked in the morning. He covered this. So one approach is to consider that all of this is a bunch of rare disorders. We need to understand their biology, and we need to have targeted treatments for the rare conditions. The other possibility is to think about things that are maybe more common or downstream targets. Remember, ibuprofen does not have any specificity for a specific biological mechanism that has to do with a particular infection that causes fever. It doesn't treat your infection, but it still has a very large effect on people's lives. So I'm in pain and I take ibuprofen, I feel so much better. So there is the possibility to treat underlying conditions, and by treat we can qualify that, right? These are developmental conditions. The difference is already there in the brain from fetal life. We're talking about changing function in a way that improves quality of life and distress. We're not talking about treating the difference. It's not, actually doesn't make sense. But we can also talk about things that may go for downstream targets and improve quality of life overall. So you can think about going after downstream targets of the many, many genetic variations that Joe was talking about this morning. There are some favorite pathways there that some of these mutations link to, like synaptic plasticity and EI balance, although I hate the term because everybody means something else with it. You can talk about methylation. You can talk about other genetic mechanisms that may be druggable. You can take some of the neuropath data. This is the one I'm the least confident about because, as you know, people don't die of their autism. So the signature of the cause of death is on the brains that we have studied, and so these are highly confused signatures. But microglia activation has been one of the most consistent findings across the small number of brains that we have access to, and so lots of people are interested in microglia activation as a therapeutic target. I'm personally a little bit cautious in a sense that, remember, microglia does not start as an immune cell. It has other functions early on in development that include pruning and making sure that your connections are efficient. And so whether microglia activation is a leftover marker of an abnormal early process or whether it's truly an immune activation is anybody's guess as far as I'm concerned. And so I'm a bit cautious about going after the target if it is not necessarily associated with distressing symptoms. Remember, the only purpose here is to treat distressing symptoms and improve function. So if this is a marker of the original condition but doesn't actually correlate with the distress you're experiencing now, why would you treat it, right? So these are things we have to figure out. The third category is it may or may not be related to the original etiology, but maybe it's an opportunity for benefit. And this is the story of the common variants, right? So a lot of the common variants probably would not cause autism, but they may modify severity. And if they're easy to go after, why wouldn't you go after them to improve some symptoms and function and some quality of life? And this is where the oxytocin story mostly fits, although there are some genetic mutations and differences, and you saw an example in the morning where you can predict actually abnormal oxytocin function in the brain. And then the last category is what we usually do, which is we treat all these associated symptoms. So the only comment I'm going to make about that category, I'm going to give you a couple of examples for the others quickly, but the only comment I'm going to make about this category is that we may have that wrong also, right? So what we do now is we say, okay, a child has autism and anxiety. I know what to do about anxiety. Let me treat the anxiety. And that's a very good thing. I think kids should be treated for the co-occurring conditions they have. But if up to whatever, 70-80% of kids, when they're 16, cross clinical cutoffs for anxiety disturbing symptoms that related to anxiety that are disturbing to them, it is possible that the anxiety is part of the original biology that produces the syndrome and not necessarily the identical thing with somebody who doesn't have autism and have anxiety. So it is important for us to figure out when the symptoms are co-occurring, because they're common, and of course they're going to co-occur, and when they're a marker of a special biological category. So a kid with autism and anxiety may have a different biology than a kid who has autism and ADHD. A kid who has epilepsy and ADHD and autism probably have a very different EI situation than a kid who has severe intellectual disability and autism but no externalizing behaviors, right? So there could be markers of different biology, and that's why you guys know that in clinical trials of co-occurring symptoms, our success rates are below what's expected for kids who don't have autism and have that condition. And there are lots of examples if you have questions about that. But let me show you just a couple of very quick ones. I know we're already quite late, right? Oh my God, okay. 2 minutes, I promise. So we took a couple of compounds from other conditions that affect EI balance. And again, EI balance means many different things to many different people. In humans, EI balance speaks to network properties. This is not the electrophysiology that you basic biologists do. So we have to be careful when we talk to each other that we don't think that we're talking about the same EI balance, because in fact it could be going the different direction, if you like, when you think about network properties. So we measure EI balance by doing MEG and EEG, and it's a network characteristic. But anyway, riluzole is a compound that's on the market for Lou Gehrig's disease or ALS. It is being used because of high EI balance in the periphery, in the motor neuron junction. But it is highly brain penetrable. So the question is, if it's highly brain penetrable, could we get some advantages from this medication in the CNS? We have tons of safety data across hundreds and hundreds of patients who have been treated with other conditions. In fact, it has been treated as an adjunct, has been used as an adjunct in OCD with some benefit in adults. And so we thought we would put it, we would do a randomized control trial in kids with autism and see whether we get any benefit. So the first thing I'm going to say about this is, for the basic scientists in the room, when you see us fail human trials, you should be cautious to interpret them as the mechanism failed. There is all kinds of reasons why we fail. The most common one is that we don't know how to measure the important stuff. So we have huge measurement problems so our measures are very, very noisy, and they're very confusing constructs. What does it mean to have social dysfunction? A million different things can qualify as social dysfunction that have very different biologies. We use a single instrument, of course we have huge amount of variability. So we have to fix our problems of measurement on the clinical side, but I'll show you just a couple of examples from phase 2 studies. So this is riluzole, this is social withdrawal from the ABC, and you'll see there is no separation, statistically significant separation between placebo, which is in red, and drug that's in blue, but there is a very consistent pattern of difference across. People were treated to up to week 12, then we withdrew the drug, and then they maintained the effect that they had, which is kind of interesting to think about in the context of an EI manipulator. Now, my error bar here is huge. So back to Joe's point, maybe if I knew what I was putting in the trial, what the kids had, I'd be able to clean this up. But it may not always matter. So for social, it probably matters. So you may want to be splitter for social. But let me show you what happens when you look at irritability and hyperactivity. My error bar shrinks right down, and in fact I have statistically significant separation nominally without multiple comparison correction. for irritability and for ADHD. So for different domains, splitting versus lumping may be slightly different in terms of thinking about how much noise does the variability actually introduce in treatment response. Sometimes it may be worth figuring it out, and sometimes it's probably okay not to, if we need to get to things that the people find useful. The second one was tidaglosib, which is a drug that did not make it into the market. It's a GSK3 beta inhibitor. It has all kinds of properties of interest that are listed there, in addition to synaptic plasticity. There was some animal model work in Fragile X to show partial rescue of the phenotype. It had been tried in dementia. It had failed in dementia, so it had died a painful death. But the license existed somewhere, and there was a small company that was interested in purchasing it, and it has been already mentioned today by somebody else. So this is not AMO1, this is AMO2. So again, when you look at social withdrawal, you'll see these huge error bars, but you see this consistent pattern, again, of separation. And in fact, if you just look at percent change from baseline, you get a statistically significant separation. If you look at repetitive behaviors on this one, again, the total score, so all these different repetitive behaviors, we're pretending it's one thing, from like ritualistic behavior to stereotypy to insistence on sameness. And there's no way they map onto the same circuit in the brain, but we pretend they do. So you get the noisy signal, but you get this kind of pattern. But if you clean it up and you look at restricted behaviors, for example, insistence on sameness, you get a very nice separation between placebo and active group. I was going to talk about oxytocin, but I'm out of time, so I'm going to skip it and I can answer questions to that. In terms of next steps, you guys talked about networks, and we're going to talk about tomorrow also, but the PON network now, which is a Canadian network, is partnering with AIMS2, which you guys also have a relationship with. And we picked up a GABA-B agonist that had failed before for reasons that we think we understand. So we are running a randomized control trial in Canada. They ran a randomized control trial on the European network. We aligned our protocols, and we have a pre-specified pool analysis at the end that the regulators accepted, so we can answer questions about whether overall it works. But then we also whole genome sequence everybody. We have EEG on everybody, and we have a sensory paradigm that's supposed to map to GABA-B to look at the subgroups that may be of relevance to a GABA-B agonist across the two networks. So thinking both on the lumping and on the splitting of this one. If it's Advil, it won't matter if we split. If it's a downstream enough target, that helps. But if it's not Advil, then we need to figure out which groups are the responders. Co-clinical trials is something that we do. I have no data to show you, but the idea here is, again, to figure out how to split and lump. And this is an example from cancer. I think it's lung cancer, where a MEK inhibitor had about a 10% remission rate. But when people tested that in mice with known genomic mutations of their tumors, you either had a 100% response rate or you had a 0% response rate. There is no such thing as a 10% response rate. You just need to know who you're treating. And if you know who you're treating, it's a much cleaner solution, right? So I'm out of time, but I was asked by some of you to say something about the Neurodevelopmental Disorders Priority Partnership. So this network is sitting as a translational network in the first valley of death, right? From basic science to actual showing clinically meaningful outcomes. But there is a second valley of death that goes from showing something to be clinically useful to actually implementing it and showing it in a big scale that it's working, that it's relevant, that people care about it, that we're not imposing our will about what we think is important to the people, what it actually matters, and so on. So you can use a network like that, although this is not our primary purpose, to think about how you can influence the second valley, translational valley. And I think you guys have referenced this yesterday. I heard Connie talked about it. So Connie, who I think for those of you who were there yesterday spoke, is the PON network is co-designed with families and people with ASD and other developmental conditions. And so one of the priorities that the families, the stakeholders, decided to get involved in is a research priority of the outcomes that actually matter to people. So I'm happy to talk more about the methods tomorrow if you like, but we now have a top 10 set of priorities for Canada. We got something like more than 1,000 questions that had to get down to a top 10. So it took us a year and standard methods to get there. So I don't want to tell you it's trivial or that it makes sense the first time you look at the data, but you can get there, and there is standard methodology for that if you're interested in it. But what's interesting here is that there's probably specificity to the local setting. So I can tell you that the priority that happened in Canada is about 50% overlapping with the priority that happened in the UK. Same populations, right? So in Canada, what everybody agreed that they wanted to prioritize was emotional regulation, anxiety, irritability, aggression, social skills, and some support for families. But in the case of the Canadians, they wanted precision treatments, they wanted biologics, and they wanted to know when, what, for how long to treat, what's the appropriate dose, what's the appropriate time. Some precision. They prioritize navigation of systems. Again, probably a local problem that needs local solutions. And they also prioritize treatments for the family. So not just support, but treatments for siblings and parents to improve the well-being of the family. So you probably need your local solution because, people with autistic people or people with autism and people with ADHD in this case, and because we had a mixed group of neurodevelopmental disorders, are as different as people who don't have neurodevelopmental disorders, and depending on where you are, their priorities may be variable. So I'm going to stop. I know I went way over. It takes a village to do this. I haven't even put names. This is like our annual Science Day, and tons of funding to get through. But I'll stop here, and I don't know if there is the opportunity to take a question or whether we're totally done.